Good evening, everybody. I'm Dr. Bobby. On behalf of New Age Cosmetic White EdTech Healthcare Consultancy, I welcome all of you back to the daily lectures. Excuse me. Today we are starting on Unit 4 of the CSIR, Life Sciences in Biology. Okay? And Unit 4 would be about cell communications and signaling. The topics covered under this would be about most parasitic interactions and recognition, and subs are there. And there would be cell signaling, then cellular communication, cancer, and innate and adaptive immune systems, right? Let me repeat that for you. Innate and adaptive immune systems. Right, we start off with the general principles of cell communication today. Right, so the mechanisms enabling one cell to influence the behavior of another almost certainly existed in the world of unicellular organisms long before multicellular organisms appeared on Earth. Evidence comes from studies of present-day unicellular eukaryotes, such as yeast, right? And although these cells normally lead independent lives, they can communicate and influence one another's behavior in preparation for sexual mating. In the budding yeast, Saccharomyces cerevisiae, for example, when a haploid individual is ready to mate, it secretes a peptide mating factor that signals cells of the opposite mating type to stop proliferating and prepare to mate. The subsequent fusion of two haploid cells of opposite mating types produces a diploid cell, which can then undergo meiosis and sporulate, generating haploid cells with new assortment of genes. Right, and the budding yeast cells respond to mating factor A. The cells are normal spherical. B, in response to mating factor secreted by neighboring yeast cells, they put out a protrusion towards the source of the factor in preparation for mating. And studies of yeast mutants that are unable to mate have been identified as they've identified many proteins that are required for a signaling process. Right, and these are required in the signaling process. These proteins form a signaling network that include cell surface receptor proteins, GTP binding proteins, and protein kinases, each of which has close relatives among the proteins that carry out signaling in animal cells through gene duplication and divergence. However, the signaling systems in animals have become much more elaborate than those in yeasts. And extracellular signal molecules bind to specific receptors. Yeast cells communicate with one another for mating by secreting few kinds of small peptides. In contrast, cells in higher animals communicate by means of hundreds of kinds of signal molecules. These include proteins, small peptides, amino acids, nucleotides, steroids, retinoids, fatty acid derivatives, and even dissolved gases such as nitric oxide and carbon monoxide. Most of these signal molecules are secreted from the signaling cell into the extracellular space by exocytosis, and it will be discussed in chapter 13 later. Others are released by diffusion through the plasma membrane, and some are exposed to an extracellular space while remaining tightly bound to the signaling cell surface. Now, regardless of the nature of the signaling, the target cell responds by means of a specific protein called a receptor, which specifically binds the signal molecule and then initiates a response of the target cell. The extracellular signal molecules often act at very low concentrations, typically less than or equal to 10 raised to minus 8 m. And The receptors that recognize them usually bind them with high affinity constant of K alpha greater than or equal to 10 raised to 8 liters per mole. In most cases, these receptors are transmembrane proteins on the target cell surface. When they bind an extracellular signal molecule, a ligand, they become activated and generate a cascade of intercellular signals that alter the behavior of the cell. In other cases, the receptors are inside the target cell and the signal molecule has to enter the cell to activate them. This requires that the signal molecules be sufficiently small and hydrophobic to diffuse across the plasma membrane. The binding of extracellular signal molecules to either cell surface receptors or intracellular receptors. Most signal molecules are hydrophilic and are therefore unable to cross the plasma membrane directly. Instead, they bind to cell surface receptors. Right? Extracellular signal molecules can activate the short or long distances. Many signal molecules remain bound to the surface of the signaling cell and influence only cells that contact it. Signaling is especially important during development and immune responses. In most cases, however, signal molecules are secreted. The secreted molecules may be carried far afield to act on distant targets, or they may act as local mediators, affecting only cells in the immediate environment of the signaling cells. This latter process is called paracrine signaling. For paracrine signals to be delivered only to their proper target cells, the secreted molecules must not be allowed to diffuse too far. For this reason, they are often rapidly taken up by neighboring target cells, destroyed by extracellular enzymes, or immobilized by the extracellular matrix. Forms of intercellular signaling. A. Contact-dependent signaling requires cells to be in direct membrane-to-membrane contact. And B. Paracrine signaling depends on signals that are released into the extracellular space and act locally on neighboring cells. For a large, complex multicellular organism, short-range signaling is not sufficient on its own to coordinate the behavior of its cells. In these organisms, sets of specialized cells have evolved with a specific role in communication between widely separate parts of the body. The most sophisticated of these are nerve cells or neurons, which typically extend long processes axons that enable them to contact target cells far away. When activated by signals from the environment or from other nerve cells, a neuron sends electrical impulses, action potentials, rapidly along its axon. When such an impulse reaches the end of the axon, it causes the nerve terminals located there to secrete a chemical signal called a neurotransmitter. These signals are secreted at specialized cell junctions called chemical synapses, which are designed to ensure that the neurotransmitter is delivered specifically to the postsynaptic target cell. The details of the synaptic signaling process are discussed in chapter 11. Right, synaptic signaling, let's see what that means. The glossary of it states that synaptic signaling is a type of cell-to-cell communication that occurs across chemical synapses in the nervous system. Synaptic vesicle would be a small neurotransmitter-filled secretory vesicle formed at the axon terminals of nerve cells and whose contents are released into the synaptic cleft by exocytosis when an action potential reaches the axon terminal. Synaptonemal complex is a structure that holds paired chromosomes together during the prophase 1 of meiosis and promotes genetic recombination. Right, so we go back to our subject proper. Courtesy to the National Library of Medicine, okay, from where we've been able to access these invaluable notes, and courtesy to the National Center for Biotechnology Information in the United States. Thank you. So I hope everybody is being benefited. Everyone reading this or listening to my lectures are benefited. Thanks again to all of them. And to follow up from where we stopped on the signaling aspects of what's called the synaptic signaling process. A second type of specialized signaling cell that controls the behavior of organisms as a whole is an endocrine cell. These cells secrete the signals, and these cells secrete the signal molecules called hormones into the bloodstream, which carries the signal to the target cells distributed widely throughout the body, right? And the mechanism that allows endocrine cells and nerve cells to coordinate cell behavior in animals is compared in figure 15.5. Because endocrine signaling relies on diffusion and blood flow, it is relatively slow. Synaptic signaling, by contrast, can be much faster as well as more precise. Nerve cells can transmit information over long distances by electrical impulses that travel at rates of up to 100 meters per second. Once released from a nerve terminal, a neurotransmitter has to diffuse less than 100 nanometers to the target cell, a process that takes less than a millisecond. Another difference between endocrine and synaptic signaling is that whereas hormones are generally diluted in the bloodstream and interstitial fluid, and therefore must be able to act at very low concentrations, typically less than 10 raised to minus 8 m, neurotransmitters are diluted much less and can achieve high local concentrations. The concentration of acetylcholine in the synaptic cleft of an active neuromuscular junction, for example, is about 5 into 10 raised to minus 4 m. Correspondingly, neurotransmitter receptors have a relatively low affinity for their ligand, which means that the neurotransmitter can associate rapidly from the receptor to terminate response. Moreover, after its release from a nerve terminal, a neurotransmitter is quickly removed from the synaptic cleft, either by specific hydrolytic enzymes that destroy it or by specific membrane transport proteins that pump it back into either the nerve terminal or neighboring glial cells. The synaptic signaling is much more precise than endocrine signaling, both in time and in space. Figure 15.5 shows the contrast between endocrine and synaptic signaling. In complex animals, endocrine cells and nerve cells work together to coordinate the diverse activities of the billions of cells, whereas different endocrine cells must use different hormones to communicate more. The speed of a response to an extracellular signal depends not only on the mechanism of signal delivery, but also on the nature of the response in the target cell. While the response requires only changes in proteins already present in the cell, it can occur in seconds or even milliseconds. When the response involves changes in gene expression and the synthesis of new proteins, however, it usually requires hours in respect of the mode of signal delivery. Autocrine signaling can coordinate decisions by groups of identical cells. All of the forms of signaling discussed so far allow one cell to influence another. Often the signaling cell and target are all different cell types. Cells, however, can also send signals to other cells of the same type as well as to themselves. In such autocrine signaling, a cell secretes signal molecules that can bind back to its own receptors. During development, for example, once a cell has been directed along a particular pathway of differentiation, it may begin to secrete autocrine signals to itself that reinforce this developmental decision. Autocrine signaling is most effective when performed simultaneously by neighboring cells of the same type, and it is likely to be used to encourage groups of identical cells to make the same developmental decisions. Thus, autocrine signaling is thought to be one possible mechanism underlying the community effect that is observed in early development, during which a group of identical cells can respond to a differentiation-inducing signal, but a single isolated cell of the same type cannot. So shown in Figure 15.6. Now, autocrine signaling. A group of identical cells produces a higher concentration of secreted signal than does a single cell. When this signal binds to a receptor on the same cell type, it encourages the cells to respond coordinatedly as a group. Unfortunately, cancer cells often use autocrine signaling to overcome the abnormal control on cell proliferation and survival that we discuss later by secreting signals that act back on the cell's own receptors. Cancer cells can stimulate their own survival and proliferation and thereby survival and proliferate in places where normal cells of the same type could not. However, this dangerous perturbation of normal cell behavior comes about, as discussed later. Gap junctions allow signaling information to be shared by neighboring cells. Right, now a second type of specialized signaling cell that controls the behavior of organisms as a whole is an endocrine cell. These cells secrete their signal molecules called hormones into the bloodstream, which carries their signals to target cells distributed widely throughout the body. The mechanisms that allow endocrine cells and nerve cells to coordinate cell behavior in animals are compared in Figure 15.5. Because endocrine signaling relies on diffusion and blood flow, it is relatively slow. Synaptic signaling, by contrast, can be much faster, as well as more precise. Nerve cells can transmit information over long distances by electrical impulses that travel at rates of up to 100 meters per second. Once released from a nerve terminal, a neurotransmitter has to diffuse less than 100 nanometers to the target cell, a process that takes less than a millisecond. Another difference between endocrine and synaptic signaling is that whereas hormones are greatly diluted, the bloodstream and interstitial fluid therefore must be able to act at very low concentrations, typically 10 raised to minus 8 moles, I guess. Neurotransmitters are diluted much less and can achieve high local concentrations. The concentration of acetylcholine in the synaptic cleft of an active neuromuscular junction, for example, is about 5 × 10 raised to minus 4 m. Correspondingly, neurotransmitter receptors have a relatively low affinity for the ligand, which means that the neurotransmitter can dissociate rapidly from the receptor to terminate a response. Moreover, after its release from a nerve terminal, a neurotransmitter is quickly removed from the synaptic cleft either by specific hydrolytic enzymes that destroy it or by specific membrane transport proteins that pump it back into either the nerve terminal or its neighboring glial cells. Thus, synaptic signaling is much more precise than endocrine signaling, both in time and in space. The contrast between endocrine and synaptic signaling: in complex animals, endocrine cells and nerve cells seem to work together to coordinate the diverse activities of the billions of cells, whereas different endocrine cells must use different hormones to communicate. The speed of response to an extracellular signal depends not only on the mechanism of signal delivery but also on the nature of the response in the target cell, where the response requires early changes in proteins already present in the cell. It can occur in seconds or even milliseconds. When the response involves changes in the gene expression and the synthesis of new proteins, however, it usually requires hours respective of the mode of signal delivery. Autocrine signaling can coordinate decisions by groups of identical cells. All of the forms of signaling discussed so far allow one cell to influence another. Often, the signaling cell and target are different cell types. Cells, however, can also send signals to other cells of the same type as well as to themselves. In such autocrine signaling, a cell secretes signal molecules that can bind back to its own receptors. During development, for example, once a cell has been directed along a particular pathway of differentiation, It may begin to secrete autocrine signals to itself that reinforce this developmental decision. Autocrine signaling is most effective when performed simultaneously by neighboring cells at the same time, and it is likely to be used to encourage groups of identical cells to make the same developmental decisions. Thus, autocrine signaling is thought to be one possible mechanism underlying the community effect that is observed in early development, during which a group of identical cells can respond to a differentiation-inducing signal, but a single isolated cell of the same type cannot. Autocrine signaling, a group of identical cells produces a higher concentration of a secreted signal than does a single cell. When this signal binds back to its receptor on the same cell type, it encourages the cells to respond coordinately as a group. However, unfortunately, cancer cells that often use autocrine signaling to overcome their normal controls on cell proliferation and survival that we discuss later. By secreting signals that act back on the cell or cell's own receptors, cancer cells can stimulate their own survival and proliferation and thereby survive and proliferate in places where normal cells of the same type could not. However, in this dangerous perturbation of normal behavior, this dangerous perturbation of normal cell behavior comes about is discussed later. Gap junctions allow signaling information to be shared by neighboring cells. Another way to coordinate the activities of neighboring cells is through gap junctions. These are specialized cell-to-cell junctions that can form between closely opposed plasma membranes and directly connect the cytoplasm of the joined cells via narrow water-filled channels. The channels are now the exchange of small intracellular signaling molecules, unicellular mediators. Such as proteins and nucleic acids. The cells connected by gap junction can communicate with each other directly without having to surmount the barrier present by the intermediate plasma membrane. Figure 15.7 shows signaling via gap junctions. Cells connected by gap junctions share small nucleotides to repeat the paragraph again. Signaling via gap junctions. Cells connected by gap junctions share small molecules, including small intracellular signaling. Right? Cells connected by gap junctions share small molecules, including small intracellular signaling molecules, and can therefore respond to intracellular signals in a coordinated way. The pattern of gap junction connections in a tissue can be revealed either electrically with intracellular electrodes or visually after the microinjection of small water-soluble dyes. Studies of this kind indicate that the cells in a developing embryo make and break gap junction connections in specific and interesting patterns, strongly suggesting that these junctions have an important role in the signaling processes that occur between these cells. Mice and humans that are deficient in one particular gap junction protein connection 43, for example, have severe defects in heart development. Like the Autocrine signaling described above, gap junction communication helps adjacent cells of a similar type to coordinate their behavior. It is still not known, however, which particular small molecules are important as carriers of signal through gap junctions, and specific functions of gap junction communication in animal development remain uncertain. Each cell is programmed to respond to specific combinations of extracellular signal molecules. A typical cell in a multicellular organism is exposed to hundreds of different signals in its environment. These signals can be soluble, bound to the extracellular matrix, or bound to the surface of a neighboring cell, and they can act in many millions of combinations. The cell must respond to this babel of signals selectively. According to its own specific character, which it has acquired through progressive cell specialization in the course of development. A cell may be programmed to respond to one combination of signals by differentiating, so to another combination by multiplying, and yet to another by performing some specialized function, such as contraction or secretion. Most of the cells in a complex animal are also programmed to depend on a specific combination of signals simply to survive. When deprived of these signals in a cultural dish, for example, a cell activates a suicide program and kills itself, a process called programmed cell death or apoptosis. Because different types of cells, require different combinations of survival signals, each cell type is restricted to different environments in the body. The ability to undergo apoptosis is a fundamental property of animal cells, and it is discussed here. Figure 15.8 shows an animal cell's dependence on multiple extracellular signals. Each cell type displays a set of receptors that enables it to respond to a corresponding set of signal molecules produced by another cell. These signal molecules work in combination to regulate. In principle, the hundreds of signal molecules that animals make can be used to create an almost unlimited number of signaling combinations. The use of these combinations to control cell behavior enables an animal to control the cells in highly specific ways by using a limited diversity of signal molecules. The different cells can respond differently to the same extracellular signal molecule. The specific way in which a cell reacts to its environment varies. It varies according to the set of receptor proteins the cell possesses, which determines the particular subset of signals it can respond to, and it varies according to the intracellular machinery by which the cell integrates and interprets the signals it receives. Thus, a single signal molecule often has different effects on different target cells. The neurotransmitter acetylcholine, for example, stimulates the contraction of skeletal muscle cells, but it decreases the rate and force of contraction in heart muscle cells. This is because the acetylcholine receptor protein on skeletal muscle cells from those on heart muscle cells, but receptor differences are not always the explanation for the different effects. In many cases, the same signal molecule binds to identical receptor proteins, yet produces very different responses in different types of target cells. reflecting differences in the internal machinery to which the receptors are coupled. Various responses induced by the neurotransmitter acetylcholine. Different cell types are specialized to respond to acetylcholine in different ways. A and B. For these two cell types, acetylcholine binds to similar receptor proteins, but the intracellular For these two cell types, acetylcholine binds to similar receptor proteins. And there's a continuation. Let me see if it can be figured out. Different cell types are specialized to respond to acetylcholine in different ways, A and B. For these two cell types, acetylcholine binds to similar receptor proteins, but the intracellular signals produced are interpreted differently in cells specialized for different functions. See, this muscle cell produces a distinct type of receptor protein for acetylcholine, which generates different intracellular signals from the receptors shown in A and B and results in a different effect. The chemical structure of acetylcholine Okay, the concentration of a molecule can be adjusted quickly only if the lifetime of the molecule is short. It is natural to think of signaling systems. It is natural to think of signaling systems in terms of the change produced when a signal is delivered, but it is just as important to consider what happens when a signal is withdrawn. During development, transient signals often produce lasting effects. They can trigger a change in the cell's development that persists indefinitely through cell memory mechanisms such as those in the chapters dealing with them. In most cases, in little tissues, however, the response fades. When a signal ceases, the effect is transitory because the signal exerts its effects by altering a set of molecules that are unstable, undergoing continual turnover. Thus, once the signal is shut off, the replacement of the old molecules by new ones wipes out all traces of its action. It follows that the speed with which a cell responds to signal removal depends on the rate of destruction or turnover of the molecules the signal affects. It is also true, although much less obvious, that this turnover rate also determines the promptness of the response when a signal is turned on. Consider, for example, two intracellular signaling molecules X and Y, both of which are normally maintained at a concentration of 1,000 molecules per cell. Molecule Y is synthesized and degraded at a rate of 100 molecules per second, with each molecule having an average lifetime of 10 seconds. Molecule X has a turnover rate that is 10 times slower than that of Y. It is both synthesized and degraded at a rate of 10 molecules per second, so that each molecule has an average lifetime in the cell of 100 seconds. If a signal acting on the cell boosts the rates of synthesis of both X and Y tenfold without any change in the molecular lifetimes at the end of 1 second, the concentration of Y will have increased by nearly 900 molecules per cell, 10 into 100 minus 100. While the concentration of X will have increased by only 90 molecules per cell. In fact, after a molecule synthesis rate has been either increased or decreased abruptly, the time required for the molecule to shift halfway from its old to its new equilibrium concentration is equal to its normal half-life. That is equal to the time that would be required for its concentration to fall by half if all syntheses were stopped. Figure 15 to 10. The importance of rapid turnover. The graphs show the predicted relative rates of change in the intracellular concentrations of molecules with different turnover times when their synthesis rates are either A, decreased, or B, increased suddenly by. Let's see the continuation there. A fraction of 10. That is, the graphs show the predicted relative rates of change in the intracellular concentration of molecules with differing turnover times when their synthesis rates are either A, decreased, or B, increased suddenly by a factor of 10. In both cases, the concentration of those molecules that are normally being rapidly degraded in the cell change quickly, whereas the concentration of those that are normally being slowly degraded, green lines, as shown in the diagram here, right, change proportionally, more slowly. The numbers in blue on the right are the half-lives assumed for each of the different molecules. Just have a quick look at the photograph here. You can take a screenshot and make the necessary notes, right? Okay, going back. The same principle applies to proteins and small molecules and to molecules in the extracellular space and inside cells. Many intracellular proteins have short half-lives, some surviving for less than 10 minutes. In most cases, these are globular proteins with key regulatory roles whose concentrations are rapidly regulated in the cell by changes in their rates of synthesis. Likewise, any covalent modifications of proteins that occur as part of a rapid signaling process, most commonly the addition of a phosphate group to an amino acid chain, must be continuously removed at a rapid rate to make rapid signaling possible. This shall discuss some of these molecular events in detail for signaling pathways that operate via cell surface receptors. We'll discuss that soon, but the principles apply quite generally, as the next example illustrates. Nitric oxide gas signals by binding directly to an enzyme inside the target cell. Although most extracellular signals are hydrophilic molecules that bind to receptors on the surface of the target cell, some signal molecules are hydrophobic enough and small enough to pass readily across the target cell plasma membrane. Once inside, they directly regulate the activity of a specific intracellular protein. An important and remarkable example is the gas nitric oxide, NO, which acts as a signal molecule in both animals and plants. In mammals, one of its functions is to regulate smooth muscle contraction. Acetylcholine, for example, is released by automatic nerves in the walls of a blood vessel, and it causes smooth muscle cells in the vessel wall to relax. The acetylcholine acts indirectly by inducing the nevi endothelial cells to make and release NO, which then signals the underlying smooth muscles to relax. This effect of nitrous oxide NO on blood vessels provides an explanation for the mechanism of action of nitroglycerin. It's nitroglycerin, okay, which has been used for about 100 years to treat patients with angina, pain resulting from inadequate blood flow to the heart muscle. The nitroglycerin is converted to NO, which relaxes blood vessels. This reduces the workload on the heart, and as a consequence, it reduces the oxygen requirement of the heart. And the heart muscles. Many types of nerve cells use NO to signal their neighbors. The NO released by automatic nerves in the penis, for example, causes the local blood vessel dilation that is responsible for penile erection. NO is also produced as a local mediator by activated macrophages and neutrophils to help them to kill invading microorganisms. In plants, nitrous oxide is involved in their defensive responses to injury or infection. NO gas is made by the deamination of the amino acid arginine, catalyzed by the enzyme NO synthase, because it passes readily across membranes. dissolved NO rapidly diffuses out of the cell where it is produced and into neighboring cells. It acts only locally because it has a short half-life, about 5 to 10 seconds. In the extracellular space, before it is converted to nitrates and nitrites by oxygen and water. In many target cells, including endothelial cells, NO binds to iron in the active site of the enzyme guanylyl cyclase. Guanylyl cyclase, stimulating this enzyme to produce the small intracellular mediated cyclic GMP. The effects of NO can occur within seconds because the normal rate of turnover of cyclic GMP is high. A rapid degradation to GMP by a phosphodiesterase constantly balances the production of cyclic GMP from GDP by guanylyl cyclase. The drug Viagra inhibits the cyclic GMP phosphodiesterase in the penis, thereby increasing the amount of time that cyclic GMP levels remain elevated after NO production is induced by local nerve terminals. The cyclic GMP, in turn, keeps blood vessels relaxed and the penis erect. Figure 1511. The role of nitric oxide in smooth muscle relaxation in a blood vessel wall. Acetylcholine released by nerve terminals in the blood vessel wall activates NO synthase in endothelial cells lining the blood vessel, causing the endothelial cells to produce The role of nitric oxide, NO, is smooth muscle relaxation in the blood vessel wall. Acetylcholine released by nerve terminals in the blood vessel wall activates NO synthase in endothelial cells lining the blood vessel, causing the endothelial cells to produce NO. The NO diffuses out of the endothelial cells and into the underlying smooth muscle cells, where it binds to and activates guanylyl cyclase to produce cyclic GMP. The cyclic GMP triggers a response that causes the smooth muscle cells to relax, enhancing blood flow through the blood vessel. Right? Moreover, after its release from a nerve terminal, a neurotransmitter is quickly removed from the synaptic cleft, either by specific hydrolytic enzymes that destroy it or by specific membrane transport proteins that pump it back into either the nerve terminal or neighboring glial cells. Thus, synaptic signaling is much more precise than endocrine signaling, both in time and in space. The figure 15.5, the contrast between endocrine and synaptic signaling. In complex animals, the endocrine cells and nerve cells work together to coordinate the diverse activities of the billions of cells, whereas different endocrine cells must use different hormones to communicate more. This is the endocrine signaling, where the contrast between endocrine and synaptic signaling. In complex animals, endocrine cells and nerve cells work together to coordinate the diverse activities of the billions of cells, whereas different endocrine cells must use different hormones to communicate specifically with their target cells. Different nerve cells can use the same neurotransmitter and still communicate in a highly specific manner. Endocrine cells secrete hormones into the blood, which signal only the specific target cells that recognize them. These target cells have receptors for binding a specific hormone, which the cells pull from the extracellular fluid. B. In synaptic signaling, by contrast, specificity arises from the synaptic contacts between a nerve cell and the specific target cell it signals. Usually only a target cell that is in synaptic communication with a nerve cell is exposed to the neurotransmitter released from the nerve terminal, although some neurotransmitters act in a paracrine mode, serving as local mediators that influence multiple target cells in the area. So going back to the subject matter proper, the speed of a response to an extracellular signal depends not only on the mechanism of signal delivery, but also on the nature of the response in the target cell, where the response requires only changes in proteins already present in the cell. It can occur in seconds or even milliseconds, when the response involves changes in gene expression and the synthesis of new proteins. However, it usually requires hours, irrespective of the mode of signal delivery. Autocrine signaling can coordinate decisions by groups of identical cells. All the forms of signaling discussed so far allow one cell to influence another cell. Often, the signaling cell and target are different cell types. Cells, however, can also send signals to other cells of the same type. And as well as to themselves. In such autocrine signaling, a cell secretes signal molecules that can bind back to its own receptors during development. For example, once a cell has been directed along a particular pathway of differentiation, it may begin to secrete autocrine signals to itself that reinforce this developmental decision. Autocrine signaling is most effective when performed simultaneously by neighboring cells of the same type, and it is likely to be used to encourage groups of identical cells to make the same developmental decisions. Thus, autocrine signaling is thought to be one possible mechanism underlying the community effect that's observed in early development, during which a group of identical cells can respond to a differentiation-inducing signal, but a single isolated cell of the same type cannot. Autocrine signaling. A group of identical cells produce a higher concentration of a secreted signal than the single cell. When the signal binds back to a receptor on the same When the signal binds back to the same receptor on the same cell type, it encourages the cells to respond coordinately as a group. And for more details, just let's look at the figure. When the signal binds back to a receptor on the same cell type, it encourages cells to respond coordinately as a group. So this is the diagram on that. I suggest you take screenshots, right? Unfortunately, cancer cells can also use autocrine signaling to overcome the normal controls on cell proliferation and survival that we discuss as we go. By secreting signals that act back on cells' own receptors, cancer cells can simulate their own survival and proliferation, and thereby survive and proliferate in places where normal cells on the same type could not. Details on this will be discussed later. Now, gap junctions allow signaling information to be shared by neighboring cells. Another way to coordinate the activities of neighboring cells is through gap junctions, right? These are specialized cell-cell junctions that can form between closely opposed plasma membranes and directly connect the cytoplasm of the joint cells via narrow water-filled channels. The channels allow the exchange of small intracellular signaling molecules, intracellular mediators such as calcium and cyclic AMP, such as proteins or nucleic acids. The cells connected by gap junctions can communicate with each other directly without having to surmount the barrier presented by the intervening plasma membrane. Figure 15.7. Signaling via gap junctions. Cells connected by gap junctions share small molecules, including small intracellular signaling molecules, and can therefore respond to extracellular signals in a coordinated way. Now, the pattern of gap junction connections in a tissue can be revealed either electrically with intracellular electrodes or visually after the microinjection of small water-soluble dyes. Studies of this indicate that the cells in a developing embryo make and break gap junction connections in specific and interesting patterns, strongly suggesting that these junctions have an important role in the signaling processes that occur between these cells. Mice and humans that are deficient in one particular gap junction protein, that's connection 43, for example, have severe defects in heart development, like the autocrine signal described above. Gap junction communication helps adjacent cells of a similar type to coordinate their behavior. It is still not known, however, which particular small amount of molecules are important as carriers of signals through gap junctions, and the specific functions of gap junction communication in animal development remain uncertain. Each cell is programmed to respond to a specific combination of extracellular signal molecules. A typical cell in a multicellular organism is exposed to hundreds of different signals in its environment. These signals can be soluble, bound to the extracellular matrix, or bound to the surface of a neighboring cell, and they can act in many millions of combinations. The cell must respond to this label of signals selectively according to its own specific character, which it has acquired through progressive cell specialization in the course of development. A cell may be programmed to respond to the one combination of signals by differentiating, to another combination by multiplying, and yet to another by performing some specialized functions such as contraction or secretion. Most of the cells in a complex animal are also programmed to depend on a specific combination of signals simply to survive. When deprived of these signals in a culture dish, for example, a cell activates a suicide program and kills itself, a process called programmed cell death or apoptosis. Because different types of cells require different combinations of survival signals, each cell type is restricted to different environments in the body. The ability to undergo apoptosis is a fundamental property of animal cells, which we'll also discuss later. Figure 15.8. An animal cell's dependence on multiple extracellular signals. Each type displays a set of receptors that enables it to respond to a corresponding set of signal molecules produced by other cells. These signal molecules work in combination to regulate each cell type displays a set of receptors that enables it to respond to a corresponding set of signal molecules produced by other cells. These signal molecules work in combinations to regulate the behavior of the cell. As shown here, an individual cell requires multiple signals to survive, blue arrows, which are shown, and additional signals to divide, shown by the red arrow, or differentiate, shown by the green arrows. If deprived of appropriate survival signals, a cell will undergo a form of cell suicide known as programmed cell death or apoptosis. Right? Excuse me. In principle, the hundreds of signal molecules that animals make can be used to create an almost unlimited number of signaling combinations. The use of these combinations to control cell behavior enables an animal to control its cells in highly specific ways by using a limited diversity of signal molecules. Different cells can respond differently to the same extracellular signal molecule is the next topic. The specific way in which a cell reacts to its environment varies. It varies according to the set of receptor proteins the cell possesses, which determines the particular subset of signals it can respond to, and varies according to the intracellular machinery by which the cell integrates and interprets the signal it receives. Thus, a single signal molecule often has different effects on different target cells. The neurotransmitter acetylcholine, for example, stimulates the contraction of skeletal muscle cells, but it decreases the rate and force of contraction in heart muscle cells. This is because the acetylcholine receptor proteins on skeletal muscle cells are different from those on heart muscles. But receptors differences are not always the explanation for the different effects. In many cases, the same signal molecule binds to identical receptor proteins, yet produces very different responses in different types of target cells, reflecting differences in the internal machinery to which the receptors are bound, as shown in figure 15.9. Right? Let's see the detail there on 15.9. Different cell types are specialized to respond to acetylcholine in different ways, A and B. For these two cell types, acetylcholine binds to similar receptor proteins, but the intracellular signals produced are interpreted differently in cells specialized for different functions. This muscle cell produces a distinct type of receptor protein for acetylcholine, which generates different intracellular signals from the receptors shown in A and B and results in a different effect, the chemical structure of acetylcholine. This is the diagram, right? Take a screenshot if you may. Okay, going back to the subject proper. The concentration of a molecule can be adjusted quickly only if the lifetime of the molecule is short. It is natural to think of signaling systems in terms of the changes produced when a signal is delivered, but it's just as important to consider what happens when a signal is withdrawn. The lecture will continue in the next lecture. This will be ending shortly. We'll be giving a lecture as a continuation to this, and the next one coming up shortly, right? So stay tuned, take screenshots, make your notes for the necessary inferences, okay? Best of luck. Thank you.